Hello and welcome, everyone. Thank you for joining us for today's webinar, Less Risk, More Speed, the new blueprint for bioconjugate development, a fireside chat. I'm Heather Masaccio, your host from Outsourced Pharma. I have just a bit of housekeeping before we get started. If you have a question for our speakers or wish to share any thoughts of your own, please submit it through the Q&A box on your screen. It's located there at the bottom. Our speakers will address those questions at the end of the presentation today. If they don't get to them, no worries. We will forward those questions on to them and they can answer those offline. Now, if you run into any problems with the stream today, please try refreshing your browser first. If that problem persists, no worries. Use that Q&A tool to submit a question, and I'll help get you back on track. This event will be available on demand. You'll receive a link via email as soon as it's available. Now on to our speakers. Today, we are joined by Dr. Campbell Bunce, Chief Scientific Officer from Abzena, and Unmesh Lal, Vice President, Healthcare and Life Sciences from Frost and Sullivan. We're going to start with Unmesh to get us started. Welcome.
Thank you, Hannah, and it's a pleasure being here today, and welcome to our discussion. Just a bit of background on myself. I've held various R&D and advisory roles in the life sciences industry for over 20 years and spearhead our biopharma contract services thought leadership at Frost and Sullivan, based in Berlin. And to kick things off, let's ground ourselves in some data, because I really believe that context matters for our discussion today. What we're seeing is that the global ADC market is on track to reach approximately $22 billion in 2026. This is the result of a decade of clinical validation, regulatory momentum, and a pipeline that is genuinely the most productive in oncology right now. While the ADC market continues to expand, its growth is not uniformly accessible due to the core development challenges that we are seeing, including therapeutic window limitations, tumor heterogeneity, and linker payload optimization. These are compounded by manufacturing complexity, where conjugation represents a key constraint. Even with very well-characterized components, achieving robust, reproducible, and scalable conjugation remains one of the most technically demanding steps in biologics manufacturing. Moving on, looking at the growth rate, we're seeing a CAGR of around 23.3% through 2030, which really reflects a category that has crossed the threshold from a niche oncology tool to mainstream therapeutic modality. First, the target biology is expanding. We are no longer confined to hematological malignancies. Solid tumors like HER2-positive gastric, TROP2-expressing breast and bladder cancers are now validated ADC territory. Secondly, the chemistry is maturing as well. So novel linker payload combinations, site-specific conjugation, and improved DAR ratio control are broadening the therapeutic window. And third, this is particularly relevant to our conversation today. The outsourcing model for ADC development has really matured to the point where integrated CDMOs are genuinely de-risking the path to clinic. Since ADC manufacturing involves a number of different steps which are very interconnected across biological, chemical, and conjugation domains, every handoff between these steps is a risk event. So that's the problem that we'll be exploring a little bit more today as well. In addition to that, there are over 200 ADCs in pre-commercial development globally, and thousands more, including preclinical programs. And critically, approximately 70% of the value in an ADC program resides upstream in the development and the CMC issues. Let me unpack what that means from a practical perspective. So process failures at discovery stage, poorly designed bioconjugation chemistry, inadequate analytical characterization are not correctable at phase 3. They become program-ending events, or worse, they lead to significant delays in approvals. So the strategic imperative really is for any sponsor, both emerging biotech and big pharma, is to front load risk identification and mitigation. Design for manufacture must start at discovery, not at tech transfer. That's going to be a caveat of our discussion today as well. A directional trend that we are seeing is this shift from ADC to XDCs, bispecific ADCs, dual payload ADCs, and antibody oligonucleotide conjugates are not incremental refinements any longer. They really represent an entirely new manufacturing paradigm. Consider a bispecific ADC, for example. You now have two antibody arms, each potentially requiring different conjugation chemistries, targeting different distinct tumor antigens with a shared payload that needs to maintain therapeutic index across both targeting domains. So the analytical challenge alone, characterizing heterogeneity, confirming DAR integrity, validating immunogenicity assays, is an order of magnitude more complex than a conventional ADC. AOCs as well, they go further. Here you are conjugating a biologic antibody to a nucleic acid payload. The chemistry, the containment, and the stability considerations are fundamentally different from small molecule cytotoxic payloads. So CDMOs cannot just retool their chemistry capabilities for these modalities, and who are not being able to do that are really going to be left behind. We are aware that Abzena has direct experience with AOC programs, and Campbell shall sort of elaborate a little bit more on this in our discussion further on. One last point in regards to ADC deal making that has really accelerated significantly. We've been tracking this market for over a decade, and with large pharmaceutical companies deploying billions into acquisitions and platform deals. Most recently, we saw Gilead Sciences' acquisition of Tubulis, and I believe just yesterday, Eli Lilly's recent ADC-focused deal underscores the access to differentiated ADC technology, which is now really a strategic priority, particularly around linker payload innovation and next-generation conjugation platforms. Moving on, what does this mean in terms of growth? We firmly believe that these bottlenecks are really creating opportunities both for the pharmaceutical and CDMOs alike. The bottlenecks in the pipeline define which CDMO capabilities become premium. The bottleneck is at conjugation, at linker payload chemistry, and at the analytical layer that bridges biology and chemistry. AOCs and ARCs, as I mentioned earlier, are the frontier here moving forward. The CDMOs that have invested in these capabilities ahead of the commercial wave are now really positioned to capture a disproportionate share of that demand. There is also a dimension from a geographical and a geopolitical perspective as well. To circumvent this geopolitical risk, the trade tension, the export control uncertainty is really accelerating demand for resilience, regional diversification in terms of manufacturing. And now what we are seeing is that sponsors are not only looking at dual sourcing, but preferring CDMOs and partners which have a US and a European platform base and also established FDA and EMA sort of track records. Now, what does this mean for CDMOs? The 5 strategic imperatives that we have identified is that fragmented vendor models are becoming obsolete. Separate antibody CDMOs, separate payload chemistry partners, separate conjugation specialists are really compounding the risk at every handoff. Each tech transfer is a potential source of process deviation, timeline delay, and data loss. Our analysis of program failures consistently shows that handoff friction is among the top 3 causes of CMC delay. The one partner antibody to vial model is not just a commercial proposition, it is a risk mitigation strategy. I think this is, we've heard this echoed through most of the sponsors that we've engaged with. When a single partner holds the scientific accountability across the entire value chain, the incentive structure really changes. There is no process knowledge lost in translation. In addition to that, what we're seeing is that design for manufacture is really beginning at the discovery stage. If your CMC team first sees a molecule at IND-enabling studies, you have already made process-limiting decisions you cannot undo. linker chemistry, for example, that is elegant in a biochemistry lab, may be entirely incompatible with GMP scale synthesis. Antibody constructs that produce excellent in vitro data may have catastrophic aggregation profiles under GMP conditions. So the CDMOs that are really winning sponsor trust are those that deploy CMC expertise as a genuine discovery partner, contributing to these antibody engineering decisions, the linker payload selection, and are really formulating the strategy from day one. This is what I mean by the front loading of risk identification. And this is what it sort of looks like in practice. And it'll be great to hear a little bit more in this regard from Abzena as well. Another point is the execution over capacity. We firmly believe that capacity, which is necessary, but it's not sufficient, particularly when it comes to ADC development, predictability, technology, transfer quality, the analytical depth are now the decisive criteria. A CDMO that can characterize your DAR distribution at every step, validate your host cell protein clearance, and detect aggregation trends before they are manifested as failed batches, that partner is worth a lot more than one with shallow sort of analytical capability. Another interesting theme that we are seeing is mid-sized specialists are really gaining relevance. When I had initiated this benchmarking analysis for ADC CDMOs, I had sort of undertaken this task by envisioning the tier one CDMOs to be at the forefront. But what we are seeing based on sponsor feedback, that there is a structural shift in terms of the mindset and the partnering with mid-sized science-focused CDMOs are really working more proactively on these complex bioconjugate assets. Large CROs and CDMOs optimize for throughput and standardization, which works really well for monoclonal antibodies, but can be a liability when it comes to these novel bioconjugates that really require a lot of bespoke process development. Mid-sized specialists offer the scientific depth, dedicated bandwidth, and critically the ability to maintain genuine partnerships with sponsor development teams rather than processing them through a production queue. And we firmly believe that this flexibility and science depth that characterize these organizations often outperform pure scale when what you need is not throughput, but precision. And we can see this with the M&A and the licensing activity that we are seeing in 2025 and now into 2026 as well. And then a final point around life cycle accountability. Sponsors are really looking for partners who own these outcomes. That distinction has been profound. It has profound implications for both CDMOs in terms of how they're structuring their engagements, what data they're sharing proactively, and how they communicate risk as well. So life cycle accountability means being present right from lead selection through commercial manufacture with consistent process knowledge, consistent analytical methods, and also consistent regulatory documentation. When a CDMO has been with the program since pre-IND, the regulatory filing is not as challenging. With that, that is a quick snapshot on the industry from our ongoing research. What I found really compelling about Abzena's model is that it is essentially a living proof of concept for each of these 5 imperatives: 2 decades of bioconjugation expertise, a dual US-UK operating footprint, their proprietary conjugation technology, and really a service architecture that really spans across discovery through commercial GMP. Campbell, let me hand it over to you so you can walk us through how some of these talking points translate into practice.
Thank you, Unmesh, and thank you for laying the landscape out so well in such detail. So my name is Campbell Bunce. I'm the CSO at Abzena. I've been with Abzena for over 10 years. We're a business that specializes in biologics and bioconjugate drug development from concept through to GMP manufacture. Prior to Abzena, I spent 18 years or so in biotech. So really understand what the challenges are that we face within the biotech sector and how to to take molecules, take new drugs, next generation complex drugs from brain to bench, bench to bedside. And that's what we do at Abzena. So I have 2 or 3 slides just to try and summarize the landscape and the kind of challenges in a little bit more detail, from which Unmesh is taking us through. So this slide here, this captures the growing momentum in the number of ADCs that have been approved, and there's certain patterns we can see within those approved ADCs. So there's a pretty common use of auristatins, the sort of microtubule inhibitors, but we're seeing more interest, more use of the topoisomerase inhibitors. We're seeing, certainly of those that have been approved, limited conjugation around stochastic lysine and cysteine, which really belies the massive amount of development activity in the new bioconjugation platforms. And there's a number of common reported toxicities, and that draws their attention to potential areas of improvement of ADCs to help open up that therapeutic window. And of course, this is the tip of the iceberg. Underneath this, there's a huge amount of development coming through in new technologies, improved platforms, as well as manufacturing processes. So this slide here really kind of highlights that there's a lot of complexity in how we need to think about ADCs, all the different components that need to come together from early design through to manufacture. There's a lot of moving parts that Unmesh has sort of highlighted need to come together in a very coordinated way for successful development. So when we're looking at the front end, antibody discovery and optimal selection, we're looking at linker payload design in combination with the antibody, the Fab, the different sort of recombinant proteins. And then looking at the kind of therapeutic index, what is the best combination of these technologies that is most likely to lead to success, and take those forward through the manufacturing campaign. Campaign includes linker payload manufacture, it includes antibody manufacture, and it includes a combination of those in conjugation. So this looks highly complex, very scary, and I haven't even included in here the analytical method development, formulation development, etc., that needs to be included and factored into every stage. Because as Unmesh has highlighted, analytical capability is really important in the successful development of ADCs. So successful ADC development doesn't just kind of magically happen. Having access to expertise in and the ability to actually utilize the tools in a deep ADC toolbox is really important. And from the ability to consider this in a very holistic way, because oftentimes we see that developers might have an antibody and they feel that we can stick something to it and we create a new drug. Well, we know that's not true, and it eventually meets with significant pain points, downstream in terms of scale and/or safety or function, etc. So, with a deep sort of toolbox with access to all these different components, such as the antibody, the Fab, the recombinant protein part, the conjugation unit, the stabilizing molecules that we can use, the linker architectures, and the payloads, then that gives us a much better chance of being able to design the most successful ADC. Underpinning this as well is the introduction of certain platforms. If within that toolbox you have access and expertise around platforms that can actually provide solutions to successful ADC development, then that's extremely helpful. In the context of Abzena, we have ThioBridge, and this is a technology that is very flexible in terms of the drug to antibody ratio designs that we can make, but also in particular how stable those molecules are. So it's a super stable platform that is highly scalable and allows for high yielding production processes. This is something that's in the clinic just now. We're very excited to look forward to the data there. But what we found is that ThioBridge outperforms every ADC that we test it against, including more recently some data that's come through in HER2. Now that outperforms in vivo, but the complexity in ADCs is that it might not outperform in vitro. So these are things that if you've got the expertise to apply, you can preempt and consider the downstream risks that we need to understand for clinical efficacy. So this slide here shows the kind of the linear route from early design and evaluation through to manufacture. This is just for the ADC, but it captures the elements of designing, making, testing, both in vitro and vivo, and ultimately manufacturing. Now, in reality, the best CDMOs in the space can condense timelines through parallel tracking these key deliverables, these key activities, generating data in real time as well to make sure that we're making good decisions about any risks, challenges, and the best molecules to take forward, but also utilizing the platforms to take certain things off the critical path. And that's what we do at Abzena. As Unmesh has explained or described, we are a global CDMO. We have a solid footprint in the US and the UK. And this has been extremely beneficial for us because not only do we offer the expertise to integrate full development of ADCs, including the biologics piece, but we are in a great position to kind of manage a lot of the geopolitical issues that we're seeing just now on tariffs in particular. So we're seeing a lot of our sort of clients look to onshore into the US where we can better manage and control the supply chain under the one roof of Abzena. So I've got another couple of slides, but I think it's probably best that we move to the questions, considering the time.
Okay, great. Well, I'm going to pop back in. I'm going to say thank you to both of our presenters for that great presentation. And yes, this is a great time to get to our audience questions. If you have not submitted one and you'd like to, you can do so now. So let's get to our first audience question. And that question is asking, what are the most important trends shaping the ADC and broader bioconjugates market today? Who can help with this one?
I mean, I can start actually.
Sorry,
sorry, do you want me to start on Mesh?
Yeah, please go ahead.
Yeah, so I mean, I love biotech, and at Abzena we are really lucky to see, so many next generation molecules coming through, and having the kind of excitement and pleasure to work on. So I would say that what we're seeing in particular, it's kind of three areas. One is obviously the technology developments, particularly around the conjugation technologies, as Unmesh has highlighted. And this really looks at offering, or at least improving on site-directed conjugation. So it allows us to better control the drug-to-antibody ratio, improve stability, improves yields. I've mentioned the example that we have as a platform in ThioBridge, but there's many other examples that help open up the application of different cargoes as well, that help us look at different disease indications. And that's perhaps the second one, the fact that we're able to learn a lot from classical ADCs, improve stability, conjugation technologies, linker architectures, and stabilization components within that, to introduce new cargoes. And these are particularly exciting. I would say that antibody oligoconjugates are an area that's growing extremely rapidly. And this helps open up new disease indications. You know, we're seeing disease indications like muscular dystrophies, cardiomyopathies, neurodegenerative disorders. We've even seen the potential application in things like preeclampsia. So the potential is massive and enormously exciting. A third area for me, we hear a lot about AI. Now it's a little bit of a slow burn, I would say, in terms of using AI to optimally design ADCs, or to consider how to optimize processes. I mean, it's moving forward. But the exciting area, I would say, when you combine it with the new cargoes, is using AI to discover new targets for antibodies, Fabs, new targets that are involved and could be targeted to design these bioconjugates to treat or to generate better treatments for diseases that were found difficult to treat in the past. So those are areas that I think are particularly exciting.
Yeah, and to add to those, I think the AI trend is one that we're seeing in terms of the recent partnerships that we're tracking as well. I think Campbell very succinctly covered this transition from first generation to this need for precision engineering. The pipeline expansion that we're tracking very closely in terms of the XTC modalities. The one point that I would also like to highlight, just based on some of the recent discussions that we are having, is this geopolitical pressure and this sort of supply chain regionalization. And we're seeing a trend that is not just about reshoring or onshoring, but it's about strategic onshoring. So that's a really interesting trend that we're tracking in addition to that end-to-end sort of capability provision. I think we did discuss analytical sophistication being a very clear differentiator and an unmet need in the industry for biotech companies, and really this push that we are seeing in terms of the M&A activity. And we believe that that's going to continue into the year.
Okay, very good. Thank you both so much. Now, this next audience question is asking, how are sponsors rethinking CDMO selection criteria today, and which capabilities have become essential versus merely desirable? Who can start off by helping with this one?
Yeah, I think that's a really interesting question, because the CDMO selection framework, it does vary by modality, biologics versus small molecules, but it has really shifted over the past 3 years. The conversation usually used to begin with capacity. Do you have available conjugation slots? What is your containment level? Can you hit our timelines? Those remain as critical qualifiers, but now we are seeing that they're no longer sufficient. So sponsors have really learned that sometimes, though expensively, that capacity without that deep process knowledge or that analytical capability that I'm reiterating again and again, and the program continuity is really a liability. And if I have to delve a little bit deeper, some of the essential capabilities, would include obviously GMP compliant conjugation infrastructure with OEB 4 and 5 containment. We're looking at end-to-end integration across antibody production, conjugation, analytics, and drug product, which I believe is easier said than done. We have many CDMOs who do claim to have that end-to-end capability, but when we start benchmarking them across these capabilities, we see a very different story, and I can delve into that a little bit later. Regulatory track record is also obviously very critical. In addition to that, the competencies in tech transfer, I think these would be some very important ones. And then specifically for some of the newer modalities, obviously, proprietary conjugation platforms, in-house HP API, and the linker payload synthesis are ones that we're hearing a lot about, in addition to that dual sort of geographical footprint, which we think is a clear differentiator as well.
Yeah, and we're seeing, certainly in terms of how the buying behaviors of ADC developers, are evolving over time. We work with a lot of different types of clients, from spin out startups through established biotech and even big pharma. And we see that more and more they're moving towards exactly what you say, Unmesh, having the more systematic capabilities and approach to ADC development, from cell line development to make the antibody or Fab or whatever recombinant protein we're looking to conjugate, the expertise around linker payloads, and importantly the optimal route of synthesis, because we don't want to be developing a process that's extremely expensive when it comes to the linker payloads. And then, introducing and having the ability to flex around platform conjugation methods to accommodate some of the new conjugation methods and certainly the new payloads that we've been seeing. And then being able to do that allows us to look at, getting from gene to IND a lot more quickly and with a lot less risk.
Very good. Thank you both so much for that. This next audience question is asking, how is the business model for CDMOs evolving as biotechs demand earlier de-risking, faster development timelines, and more accountability across the life cycle?
Yeah, I think that's a really interesting one as well, because that is one that is interesting from a very structural shift perspective. And then when we speak to biotechs vis-a-vis big pharma, we see some slight changes in that as well, in terms of their preference for the optimal business model. The traditional model has been very transactional. Sponsor sort of pays for very defined deliverables, the GMP batches, for example, an analytical report. But that's really being disrupted by two forces, I would say. The biotech sponsors that need a lot more support when it comes to execution, and also the competitive pressures amongst CDMOs to differentiate themselves beyond just pricing. So some of the business models that we are seeing, particularly for this modality, that are coming to the forefront is this sort of shift from the transactional to the co-development partnerships. We are also seeing milestone-based business models that are being explored, very outcome sort of oriented commercial structures. We are seeing very dedicated program teams and capacity reservation as one of the strategies as well. And then, as I had mentioned earlier, being sort of a life cycle accountability partner as a differentiator when it comes to being a CDMO.
Yeah, again, talking through direct experience with our clients, what I think oftentimes we miss as a contract organization, just because we get our heads down and focused on what we've been asked to do, we forget to ask, what the ultimate goal is of our clients, and where are they actually going? Do they want to get fast to clinic to establish some early clinical data to leverage the value of their technology to out-license or be acquired? Or are they looking to build a pipeline around the platform antibody or Fab? And they're needing to figure out what the optimal kind of cadence of that is, and work with a partner to do that. So as I say, we oftentimes forget to ask that. What we're doing more, certainly at Abzena, is paying more attention to that particular aspect. What is the overall strategy of our client? What is the overall goal? And strategically, how can we support that? And towards that also, because there's a big technical element to this. And towards that, we don't have this kind of, the client has to fit into our fixed model or our fixed platforms. We're seeing clients wanting more flexibility, agility and flexibility around what we can do, particularly, as you said earlier on, Mesh, the sense or the use of data, the use of data to make decisions. And oftentimes we get data we don't like at the early parts of development. So we need to be nimble and agile enough to be able to pivot and support our client doing that. So we've introduced, what we call a sort of technical excellence oversight team. So these are seasoned drug developers, biotech experts who have come into the business, and we apply to our key integrated client programs to help do those two things, to help better understand the strategy and the ultimate goal of the client, but also then work internally with the project management, with the tech ops folks to actually deliver on that strategy.
And if I could just add to that, there was an Abzena sort of business model that really resonated well with sponsors, the grow with the client, and particularly for biotech companies, I think that's very pertinent, the challenges that they sort of face, not only from a funding perspective, but some of the pressures that they are under. So that is something that, based on our analysis, that we felt was very critical, and that's what differentiated specialist CDMOs from some of the tier one vendors.
Yeah. Yep.
Okay, very good. Thank you both. This next question is asking, for AOC, what is the proper DARs or DARs, and is there a good strategy for DAR1? What about this one? And I hope I asked it correctly, gentlemen.
So, what is the proper drug to antibody ratio or DAR for AOCs? It's one of those situations where, we're kind of stuck with the kind of significant properties of the two molecules that we're trying to bring together. So there's a net positive charge in one and there's a net negative charge in the other, and they're pretty substantial molecules. So when you're linking an oligonucleotide to an antibody, you need to account for that. So, because of those constraints, more often than not, it's DAR1. So one oligonucleotide to one antibody. As I say, principally through this physical chemical constraints. Sometimes DAR2, we've seen DAR2. And we've seen a number of different conjugation methods to manage that. So if you're using a whole antibody, you can have an engineered element into the antibody. You can have an engineered cysteine for, conjugating directly to that engineered cysteine, usually in the heavy chain, your oligo, or if it's a Fab, the truncated form of an antibody, the antibody binding region. These work really well with oligos because you can use cysteine. Our ThioBridge actually works exquisitely well with oligos because it rebridges across the disulfide bonds of the Fab and creates a really stable, highly scalable, high-yielding sort of antibody oligo conjugate. But there's different methods, and as I said earlier on, there's a lot of development in next-generation platforms, both for conjugation, but also for linker architectures that allow us to stabilize some of these more difficult cargoes that we're trying to attach to antibodies and Fabs.
Very good. Thank you so much. Now, this next audience question asks, when benchmarking CDMOs in the ADC market, what truly differentiates leading players today beyond scale or installed capacity?
I can take that one because I believe that's on the back of some of the research that we've done in-house. And we started up with a list of around 50 ADC CDMOs, and then we sort of applied some of our growth and innovation metrics to benchmark these vendors, and then also get feedback from sponsors as well. And we'd identified 16 leading ADC CDMOs globally. And the criteria that we applied that we felt were the most critical were obviously the proven commercial scale execution. We can't sort of forego that. Multi-year regulatory track record, We also sort of looked at their end-to-end capability, which is where we found the most differentiation in terms of the leaders versus some of the laggards and the emerging players as well. Their commitment from a CapEx perspective as well. So these were some of the base criteria that we leveraged. But then I think to add on to that, some of their proprietary technology platforms, and that's where things get a little bit more complicated, their analytical depth and some of their assay development solutions, I think, were also at the forefront as well. End-to-end integration, both from a biology and a chemistry perspective, were very critical, and the CMC expertise. So we had some very detailed discussions around some of these areas as well. And then finally, obviously, it's a very sticky business. So the sooner we engage on vendors engage with sponsors, the more likely they're going to have that sort of partnership, particularly with biotech companies. So customer satisfaction and repeatability of business was a very good criteria in terms of the quality signals to sort of identify the leaders in this benchmarking exercise.
I mean, yeah, I mean, obviously, this is independent sort of research, etc. I mean, I would say that what we're finding as well that's particularly popular is not just the technical strategy and the route to manufacturing and getting quickly into patients, that holistic approach and, you know, broad capability offering. The other is the kind of regulatory path, because, you know, ADCs are still relatively new to the regulators. And, you know, I think they like a very proactive approach. You know, we've got a dedicated regulatory expert on our team. But we're finding that regulators like a proactive approach, and they like to see data, particularly orthogonal data or data from orthogonal assays, not just your standard assays, that help us really understand the ADC that we've designed, you know, its properties, its stability, etc. So, you know, there's a big regulatory element there that we pay particular attention to, and we find that lands well with our clients.
Okay, very good. Thank you so much for that. This next question asks, where do you see the most compelling growth opportunities for CDMOs in the ADC and next generation bioconjugate space over the next 3 to 5 years? I can help with this one.
I mean, I can start. It's mainly to reiterate what Unmesh and we've said to a large extent already. We're seeing that people are getting, or the developers are getting very bold in the design of the molecules. You know, bispecifics in terms of the antibody binding to 2 different targets, and then combining them with dual payloads, as Unmesh spoke about earlier on, you know, thinking about how we then consider optimal conjugation platforms to make this kind of component a reality, or this ADC a reality. But we are able to do this, and we're able to do it if we are kind of clear-eyed about the upfront design and developability aspects, and not try and shortcut that particular phase of development to try and get something into the clinic as quickly as possible, something that, you know, we can't make to any particular scale, and it's never going to be a commercial product because we can only make, you know, a few grams of it and not the kilograms that we need to make to satisfy the patient population. So a lot of great designs, but obviously that opens up, well, for cancer, that's great, but it opens up a number of different disease indications of unmet need, which is extremely exciting from my perspective.
Yeah. And again, I think just to add on to that, the next-gen modalities, AOC, I think ARC is a very interesting area as well that we're tracking closely. We are seeing the whole value proposition in terms of antibody to vial, the platforms and which CDMOs and how they're sort of either partnering or building those capabilities to sort of capture that downstream value. That's an opportunity that we're tracking very closely. This trend that I mentioned earlier in terms of the regional hub development, you know, just to overcome some of this issue with resilience and some of the supply chain challenges, which we believe in the next 3 to 5 years, you know, is really going to come into play. And then one last point that I think is we're having conversations around this, but obviously the industry has been a bit of a laggard in terms of adoption, is obviously the use of digital twins in manufacturing. So this is an area that we are also tracking from a digital perspective as well.
Okay, excellent. We have a question come in about asking about typical costs for AOC. Is that something that you can touch upon in this presentation?
It depends. I mean, there's a lot underneath that question. You know, what is the antibody? Is it a bispecific? Is it, you know, a classical or standard, you know, antisense oligo, or is it siRNA, etc. So I wouldn't want to offer a cost on air, as it were, but certainly happy to receive more information about the particular AOC that you would like to have designed, and we can talk about cost then.
Okay, very good. Sorry, Enmesh, go ahead.
That is a very tricky question. We do put together some cost estimates when we are sizing these markets, but that is a very sticky topic when we're talking to CDMOs in regards to their pricing. But if I had to give a range, for example, you know, it's obviously way more expensive when it comes to AOCs, tens of millions in terms of infrastructure, in the range of about 25 to 40 million upfront, and then around 45 to 65 million when it comes to annual manufacturing costs. And R&D really consists of about 30% of the total program spend. So that gives you an overall idea in terms of the range that we're looking at. But again, there's a big caveat here in terms of, you know, scale and you know, where the company is. There's a significant amount of variance in these numbers.
Yeah, and we need to factor or consider also, you know, some AOCs require high frequency of high doses. At least we're seeing that in some clinical studies for particular indications, and then others, you know, considerably less. So, you know, there's a massive variability in those overall costs. But there's no substitute for spending a little bit more early on in terms of the design developability, because that can mitigate a ton of costs downstream.
Okay, well, I think that is really our time for today. Do you have any last final parting thoughts you'd like to share with the audience before we go?
I mean, I would say that, you know, it is an incredibly exciting time for drug development. You know, we've got a platform technology that is proliferating in terms of potential and in terms of value to future patients. I think it was John Lambert, you know, one of the giants in the ADC field. He said, you know, last decade or so has been antibodies. This decade is ADCs. So, you know, exciting times.
I would concur. You know, I think there's a lot of excitement. However, I do want to point out we need to sort of balance that hype with what the reality actually is. And that's where we feel certain CDMOs and certain specialists are really going to be at the forefront in regards to this transformation.
Okay, very good. Well, thank you both. Thank you to our presenters for sharing their time and their expertise. And thank you to audience for tuning in. Just a reminder, this will be available to you on demand. You'll receive a link via email as soon as it's available. We'll see you next time on Outsourced Pharma. Have a great day. Take care. Bye-bye.
Thank you.